thank you, Ashley, for the introduction. Like you said, my name is also Ashley. I'm a postdoc at the Embodied Computation Group at the Centre of Functionally Integrative Neuroscience at Aarhus University in Denmark. And today I'll be giving you a tutorial about how to run dynamic causal modelling for evoked responses using the SPM12 toolbox. There we go. Okay, so just a quick overview of what I'm going to be talking about today. I'll start with a very kind of brief overview about the basics of dynamic causal modelling, and then a brief introduction into the example data that we'll be using today. I believe with the sign up link there was a link to the GitHub repository that was shared as well, and that contains all the example data. I'll just say very early on, if you're following along with the example data, you do need to download the whole repository, not just the one data file, because there's some dependencies and stuff. So I'm saying that now just in case anybody's only downloaded one file. You do need the whole repository. I'll then give a walkthrough of the DCM pipeline in the SPM user interface, and then a walkthrough of some of the options to view the results in different ways. And then if we have time at the end, I'll also cover how to run a Bayesian model selection. So the main aim of this tutorial is, at the end, we want you to be able to estimate effective connectivity between sources of neural activity in the context of a visual repetition memory paradigm. Okay, so what is DCM? Dynamic causal modelling is based on an idea that was originally developed for fMRI, in which measured data are explained by a network model that may consist of a few different sources that dynamically interact with one another. And through using DCM, we can invert this network model using a variational Bayesian scheme, and that allows us to make inferences about these different connections between sources in the network that we've defined, and how exactly these connections might be modulated in the context of, say, a behavioral paradigm. So this type of connectivity that we're talking about is known as effective connectivity, which is context dependent. So rather than structural or functional connectivity, which we've probably already heard of, DCM estimates this effective connectivity. So just to recap, we probably know what structural connectivity is. It's the physical connections between different neural structures, and we might examine this through, for example, diffusion-weighted imaging. Functional connectivity, on the other hand, more describes the statistical kind of correlation or covariation of activity between discrete sources of the brain, and we can look at this by using fMRI. Effective connectivity kind of takes one step further than functional connectivity, in that it's kind of defined as the causal influences between different neuronal populations. It describes the more contextual influence that one region of the brain may have over another, such that we can examine or interrogate these connections in a much more context-dependent fashion. All of that is to say that DCM is very much a hypothesis-driven method. When you're employing your DCM, you make a lot of decisions that are very specific to your data set and the questions that you want to ask your data. So you can use it to test these very specific questions and hypotheses about the activity between the sources in a predefined network, rather than simply being limited to asking questions about the strength of source activity on an individual level. You can use this to even compare connection strengths between healthy individuals with a patient group, for example, depending on your paradigm. Another great thing about DCM is that through this inversion that I mentioned before, according to a variational Bayesian scheme, you can yield an approximation to the log model evidence. And you can basically use this to compare alternative models of the same data. I stress the same data. When you're conducting a Bayesian model selection, the purpose of this is to compare how different models best fit the data, but the imperative thing is that you can only compare models of the same data, because obviously comparing models of different data, different models may fit different sets of data in different ways. So when we come to Bayesian model selection later on, it is comparing different models of the same data set. Okay, so through this model inversion, I've mentioned model inversion quite a few times already, even though it's only been about 5 minutes. We can invert this model, and that allows us to infer a set of neuronal parameters, which here is denoted theta, so we can approximate the posterior probability of a set of parameters theta, given our data set y on our model M. And our model may describe a set of microscale synaptic dynamics, So since MEG and EEG is much more highly resolved in time compared to fMRI, which obviously DCM was initially developed upon, we can actually use our DCMs for EEG and MEG to estimate activity on the level of single synaptic currents, if you choose to run conductance-based modelling, for example. We can also run a forward model, which allows us to predict the measured activity, so estimate the probability of a kind of simulated data set Y given a set of parameters theta and model M. So overall, what does this actually mean in terms of real data collection and analysis? Let's say that you start off with some EEG or some MEG data that you've collected on a group of participants that are completing a task. Let's say it's a visual stimulus and participants are presented with this visual stimulus, and based on that they have to make a binary decision choice. Let's say they're shown an image and you have to decide whether they've seen this image before or not, and it's a yes or no, and you have this button press. And then you look at the sensor level data. You have two conditions, so condition one being images that they haven't seen before, and the second condition being images that they have seen before. And you can look at the ERPs that are produced on a single channel level and compare how those ERPs look in the different conditions. Now, let's say, for example, that you have healthy controls and you want to compare that to a patient cohort that are completing the same task. And you could look at the ERP level data from that single channel, and you can compare the two between the healthy controls and the patient, and you can say, okay, there's some differences between the two conditions. Those differences are greater in the patient population compared to the control. The deflections in the, I don't know, N100, P200, whatever, it's stronger in the controls compared to the patients. That's very nice. Some people want to do that, that's fine. But what does that actually mean? But purely examining the data on the sensor level, it doesn't really give us a lot of insight into what's actually happening in the underlying neuronal processes. Like what are actually the neural mechanisms that are doing to cause these differences in these ERPs. And that's where dynamic causal modelling comes in. So what we can do is we can map our EEG channels in 3D space in the brain using, excuse me, using 3D spatiotemporal source reconstruction, which estimates the distribution of cortical sources that may give rise to these EEG signals, these ERPs that we observe at the sensor level. And we can use this to infer a network of active sources that may generate these ERPs. And we can use DCM to do this. So DCM estimates these parameters both within and between sources of activity in the network that we've defined. So we can examine these connections both in the context of regional activity changes and also in a task-dependent fashion. So you can really derive the way in which any of these experimental conditions that form part of your task either recruit or modulate any specific connections in your network. Okay, so that was a whistle-stop tour of DCM. This is the paper that we'll be using the example data from. So it's lateralized memory circuit dropout in Alzheimer's disease patients. Essentially, there was we examined the recognition memory capacity of healthy aged controls compared to Alzheimer's disease patients. In this paper, participants completed two different types of memory tasks. One was an implicit memory task and one was an explicit memory task. The example data we'll be using today is just from that explicit memory task. Excuse me. And the key aims of this study were to investigate how interregional connectivity and within-region dynamics during these two implicit and explicit memory paradigms may be implicated in Alzheimer's disease, and to really examine whether these connections in one hemisphere may provide compensation over the other hemisphere during these memory tasks. And we hypothesized that hierarchical left hemispheric specific connections may be weakened in the Alzheimer's patient cohort compared with healthy age controls. So a quick overview of the task itself. So this is the explicit memory paradigm. Participants were first underwent a training phase where they were shown 100 images of nameable objects. And this training phase took place outside of the scanner. So they were just shown these nameable objects, and let's say there was a picture of a cat or a picture of a cabbage, it's either a cabbage or a lettuce. I don't really know what that even is. I'm not so hot on vegetables, but it's a vegetable. So they were shown these images of nameable objects, and all that the participant had to do was covertly name the object that was in the image, so just name it in their head, and then press a button as they did so. So that was the training phase, and then they went into EEG scanning and were shown once again 100 images of nameable objects, but this time 50 of them were brand new images, and 50 of them were repeated from this encoding phase. And what the participants had to do was register with a button press whether they thought they had seen the image before or whether they had not seen the image before, basically. So we have two task conditions. We have the novel image condition and we have the repeated image condition. And then this is just the analysis pipeline of what pre-processing has already been done to the data that we'll be using today. So we started off with 64-channel EEG data, which was then bandpass filtered between 2 and 30 Hertz. It was epoched between -52 and 500 milliseconds peristimulus time, and it also underwent manual artifact correction. We then ran this 3D spatiotemporal source reconstruction that I mentioned earlier to identify a set of sources that we would use in our model. And we then conducted our dynamic causal modelling, and that's the section that we'll be focusing on today. Okay, so in this example for my dataset, I generated this group level source activity map using this 3D source reconstruction. This is just a one sample t-test across all participants. The reason we just ran a one sample t-test is because we wanted to use the same network model across both patients and controls to see which of the connections were either strengthened or weakened depending on the kind of patient versus control group diagnosis. So the kind of more yellow colour indicates a higher Z-score and the more red colour indicates a lower Z-score in this one sample t-test. So we identified 6 sources of activity based on this. So it was both based on the 3D source reconstruction, but also based on the relevance to the task. Based on the previous literature, what regions of the brain would we expect to come up in this paradigm, and also previous analyses conducted by Jessica Gilbert and Rosalind Moran, who originally collected the data back in Virginia Tech in the US. So if I just get my laser pointer. Yes, okay. So we identified in the left hemisphere inferior frontal gyrus, inferior temporal gyrus, and occipital gyrus, and then pretty much the same in the right hemisphere as well. Right occipital pole, right inferior temporal gyrus, and right inferior frontal gyrus. So the question now is, how does this exactly translate into a model that you're going to put into SPM? So what I find is always useful to do is kind of draw this out as a graph, basically. These are just the 6 sources drawn out as a graph. We have on the left side, we have the left hemispheric sources, starting with the occipital gyrus, temporal gyrus, and frontal gyrus, and then the same with the right hemisphere as well. Now, in my DCM, I want to define both forward and backward connections, excuse me, both forward and backward connections between these sources that are represented by the arrows here. I'm just going to take a drink, I've got a bit of a cold. So the forward and backward arrows represent these forward and backward connections between these sources. And then I also have an arrow here at the bottom which represents a subcortical input. Obviously it's a visual task, so we expect there to be some visual input into the occipital sources in the network. You can also see that I have numbered each of the 6 sources, 1, 2, 3, 4, 5, 6. You will need to do this if you have your own data set, and you want to construct your network model for your DCM on your own data set. You will need to number your sources as well, and it will become clear why in a second. Something I personally like to do, you don't have to do this, but I like to use the odd numbers for the left hemisphere and the even numbers for the right hemisphere. You don't have to do that, that's just, it helps my brain kind of mentally map where everything is, basically. Yeah, so I've just numbered these 1 to 6. So to transfer this into SPM format, you need to specify 3 different types of matrices. We have the A matrices, we have the B matrices, and we have the C matrix. So the A matrices define the connections between your specified sources. And there are 3 different A matrices. There are the forward connection A matrices, the backward connection A matrices, and the lateral connection A matrices. So for this data set, I didn't define any lateral connections. we wanted to kind of look at how different connections in the left hemisphere versus the right hemisphere changed in different contexts of the task. So that's why we didn't define any lateral connections. Our hypotheses were very much kind of wanting to look at left hemispheric and right hemispheric connections separately. So that's why we didn't define them. But we did define forward and backward connections. So in the A matrices and also the B matrices as well, the columns, if you can see my laser pointer, the columns indicate the region or the source that is the connection is coming from. And then the row indicates the source where the connection is targeting. So we can see we have a 1 here in the first column and the third row, which means that we're defining a forward connection from region number 1 to region number 3. So if I go back, region number 1 is the left occipital gyrus, and region number 3 is the left inferior temporal gyrus. So that is us defining this forward connection here from the left occipital gyrus to region number 3, the left inferior temporal gyrus. I hope that makes sense. So then we define the remaining forward connections from the right occipital pole to the right inferior temporal gyrus, and then similarly again for the left and right temporal regions and the left and right frontal regions. Similarly with the backward connections, we have a backward connection from column 3, so from region 3 to region 1, because it's row 1. So this is a backward connection from the left inferior temporal gyrus to the left occipital pole. It is confusing the first time that you do this. This is why I always like to draw it out as a graph like this and then number the sources in this way, because trying to go from this sort of thing straight into the matrices is an absolute nightmare. So draw it out, number your sources, and then transfer it to matrices like this. These are the A matrices, and I'm defining those connections there. Then we also have the B matrix, which is the modulatory connections. So it's very similar to the A matrix, but these connections in the B matrix are defined in a context-dependent manner. So it's how much these connections modulated by your task condition. So for the modulatory connections, I've just identified all of the forward and backward connections I've already defined in the A matrix, and then also self-connections which are on the diagonal. The reason I haven't defined self-connections on the diagonal for the A matrices is because that is already in the model. When we come to looking at MATLAB, you will see that the radio buttons for that options are blanked out, and that's because they're already in the model. Self-connections are automatically put in. And then finally we have the C vector. So the C vector defines the sources that are receiving any sort of subcortical input. So if we go back to our graph, we said that we want subcortical input into the left and right occipital regions, which is basically visual cortex, which in the context of our task makes sense. We want to model the visual input that is coming in as a result of the visual presentation of the stimuli. So we've added a 1 in position 1 and 2 in that vector to represent those sources. Yes. Okay, so once we've defined those aspects of the model, we can then invert our model, like I've mentioned before, to estimate the posterior probability of our set of parameters theta, given the observed data and the model M. And when we're choosing which kind of DCM we want to run, that once again really depends on the questions that you're asking the data and what kind of paradigm you've used. So you can either go with a kind of linear second order model or a convolution-based model. So for example, a canonical microcircuit, and that kind of primarily considers the cortical column. So if your hypotheses are kind of more, you want to model, excuse me, you want to model things in a more kind of a predictive coding fashion, you might want to go with a canonical microcircuit model, or alternatively, you might want to go with a conductance-based model if you want to model the electrophysiology of the single cells. And like I said, this choice really depends on the type of inference that you require for your hypotheses. For this data set, I'm going to use a conductance-based neural mass model, specifically the NMDA model, which includes a model of the NMDA receptor by utilizing dynamic Morris-Lacar type nonlinear differential equations, which describes the flow of ions at the synapse. And these equations specifically model 3 separate cell subpopulations. So we have excitatory spiny stellate cells that are in the granular layers of the cortex, and then it also models inhibitory interneurons and pyramidal cells in the extragranular layers. And these cell subpopulations are all interconnected both within sources by intrinsic connections and also between sources by extrinsic connections. And you can also expand this equation to describe single cell currents in each of these different cell subpopulations. So just to talk through a couple of the parameters here, we have gamma 3,2 here. So gamma parameterizes the connection strength between the cellular layers. So we have gamma 3,2, which means that it's parameterizing an excitatory connection from cell subpopulation 3, which is our pyramidal cells, to cell subpopulation 2, which is our inhibitory interneurons. And although connections between the within region cell subpopulations are denoted gamma here, the between region connectivity, so the forward and backward and lateral connections that I've just mentioned, they take the same mathematical form as the within region connections, and that's what's going to form our A matrix, basically. Okay, so a quick summary of what we actually found in our study. So after running our DCM, we found that there was a significant downregulation of temporal frontal connectivity from the left inferior temporal gyrus to the left inferior frontal gyrus in patients compared to controls, as well as a significant downregulation in the subcortical input into that left visual cortex in patients compared to controls. And what we also found is a significant upregulation in controls in the temporal occipital connectivity in the right hemisphere, and that had a significant correlation with task performance. So we saw this significant dropout in the left hemisphere and a potential upregulation or driving of compensatory mechanisms in the right hemisphere. So, okay, I'm going to stop showing my screen and we're going to go to MATLAB. How on earth do I do this? Okay, I think I've stopped sharing my screen. I'm now going to attempt to share MATLAB. Let's see how that goes. Okay. Okay, perfect. Okay, so I've opened the MATLAB window. I hope you can see it. Now what I'm going to do is I'm going to open SPM. So instructions on how to open SPM are already in the installation guide that got sent out with the registration link for this talk. But in the command window, I'm going to type SPM EEG and press enter. And this is going to open the SPM user interface. Okay. So now that we have the interface, we have this beautifully stylish grey and green window. Excellent design choices here. There's titles in each of the sections which you can't really see because it's white text over a grey background. Once again, inspired design choice. But the first one says temporal pre-processing. So the first one is kind of where you do any sort of pre-processing on your data. The second one is spatiotemporal modelling, which you can do your 3D source reconstruction and run your DCM. The third one is model specification, review and estimation. And then the fourth one is inference. What we're going to do first before we start specifying our DCM is click 3D source reconstruction. And the reason for this is we're going to rerun the 3D source reconstruction. It's very quick, but the reason we need to do this is that when you run the 3D source reconstruction, there's basically an assumption of the kind of imaging solution that you're running in the source reconstruction that assumes that the sensor level activity is the result of a summation of many dipolar sources that are distributed over your cortical sheet. So it kind of estimates the sensor data in kind of a vector of source space, and then also a lead field, which is kind of the mapping from sensor space to source space. But the lead field, or the gain matrix, as it's also called, is saved as a separate file. Now, what SPM does is when you save this gain matrix from the source reconstruction, it hard codes a file path in that. So even if you've downloaded the entire GitHub repository, which the gain matrix contains, it has a hard-coded path, and it looks to one of my laptops that I had like 50 million years ago. So we're going to rerun this. It's going to create a new gain matrix file, and then that is going to have a hard-coded path to your local directory, so then it will work. So that is what we're going to do now. So in the EEG/MEG source localization window, we're going to click load. And then in data files, this is the data file that we're going to click, this MFAEFFSPMEEG example. It's a horrible file name. The reason for that is because if you do your pre-processing in SPM, every time you run a pre-processing step, it prefixes a letter to the start of your file name, which has an indication about which pre-processing step you've just conducted. So if you're using somebody else's SPM pre-processed data, you can kind of make some guesses about what pre-processing has actually been done to it based on those prefixes. And that's why it's got an absolutely cursed file name. So we've loaded that in, and now we're going to click template here. So we click template, and we can see what kind of cortical mesh that we're going to use. So we're just going to click normal. There we go. And then this is the head model, which we can move around. Those kind of pale blue dots you can see there, they are the fiducials. So when you're doing MEG or EEG, you mark the locations of the fiducial points, which are basically like right in between your eyebrows, and then on your left and right ear, where your ear kind of joins the skin of your head at the top, basically. It's mapping the center space onto a head model, basically. We're going to click co-register. There we go. I'm just going to briefly breeze over this because I'm going to focus more on the DCM today rather than the source localization, but we do need to do this to fix the file path. I'm going to click forward model, and I'm going to go EEG BEM, boundary estimation maximization, that stands for. Okay, and now I'm going to click invert. Reconstruction, I'm going to go with imaging. All conditions and trials, yes. And the model is just standard. And now we have a little progress bar. There we go. I think it's almost done. There we go. Okay. So what that has now done is it has created a new gain matrix file. So now we can run the DCM, and the DCM pipeline will be able to find that gain matrix file without freaking out about file path. So we can close that window. We can close that. And we can return to our main SPM menu, the beautiful green and grey scene, and we can click the DCM button here, which is in the spatiotemporal modelling section. Okay, and now it has opened the DCM for EEG window. Okay, so we have this section at the top, load, where we can load a pre-existing DCM. If you've made a DCM before and you want to load it back in and have a look at the parameters, you can click that. You can click save if you're working on a DCM and you want to save it under a particular file name before you run it. You can click save, but we're going to click new data because we want to load in some new data to run a fresh DCM. So I'm going to click new data, and then I'm going to click this MFAEFFSPM EEG example data prop. Okay, and there's our data. At the top here we have two different drop-down menus. On the left, we have the kind of DCM that we want to run. So we have event-related potentials, cross-spectral densities, time frequency modeling, induced responses, phase amplitude coupling, and neural field model. And we're just going to stick with ERPs because this is a tutorial for evoked response DCM. So we're going to take that. And then on the right-hand side, we have a drop-down for which kind of neuronal model we want to use. So if we want to use a convolution-based model, linear second-order modelling, or conductance-based modelling, like I mentioned earlier. So we have many different options. Once again, we have ERPs, we have sensory evoked potentials, two different types of canonical microcircuit modelling, the NMDA model, neuros mass modelling, mean field modelling, and locus field potentials. Like I said in the slides earlier, we're going to go with the NMDA model. There is a paper that was linked in the sign-up by Rosalind Moran. It was Neural Masses and Fields in Dynamic Causal Modelling, I think it's called. And it really breaks down all of these different options really beautifully. Because I've only got an hour today, I can't really go into too much detail about all of the other options. So I would highly recommend reading that paper if you have questions about any of the other types of modelling. So we're going to click the NMDA receptor here. I'm going to kind of prefix all of this analysis by saying all of these decisions that you make in this pipeline is very much dependent on your data set. It's dependent on the task that you've employed as part of that data collection, and it really depends on the specific hypotheses that you have, like what questions do you really want to ask the data. So all of the options I pick here today are very specific to this data set. When you're using your own set of data, all of these options, you will really need to think about why you're choosing those things, and kind of, it can be very much a trial and error process. And that's why we have Bayesian model selection, right? So we can compare different models with different sets of parameters and see which of those models best fit your data. So just a caveat at the beginning. So in the first section called Data and Design, we're going to define some of the parameters that describe our data set. So for the time window, I want to look at 0 to 450 milliseconds. In our between trial effects, we have two conditions. We have the novel image condition and the repeated image condition. So I'm going to type 1 and 2 there, and then I'm going to type 0 and 1 here. Now, in the event that you have multiple trial task conditions and you can't remember which order these different task conditions are saved in your data file, what you can do is you can go to the command window in the main MATLAB window, and yeah, I'm going to type d equals spm underscore eg underscore load, and then I'm going to copy in the name of the mat file of the data. There we go. We've loaded in the EEG or MEG data structure here. Now, if I type d dot, move my cursor, cond list, it's going to give me the list of conditions in my task. So I have a cell array saying novel and repeated. So I know that in the data file, the novel condition is condition 1 and the repeated condition is condition 2. So that's just in case you forget the order of the conditions, basically. Okay, now, back in the DCM window, if we're in Data and Design, I'm going to click the Display button that's here. So now we see the two different conditions. Before it was 1 because we hadn't defined the two different conditions, but now we can see the two different conditions. So observed response 1 we know is the novel image condition, and observed response 2 we know is the repeated image condition. Now we have some different options here. We have the trend, which allows us to remove any low frequency drifts at the sensor level that we might want to remove. So it's the number of discrete cosine transform terms that you would like to remove. I'm going to leave this at 1. What I like to do is, if I want to do any sort of pre-processing on the data, I like to complete that outside of the DCM pipeline and then just run the modelling here. But if that's something that you want to do, you can do that here. We also have the subsampling option, which allows us to do any further downsampling of the time bins if you want. I'm also going to leave this at the default option of 1. And then we also have the modes, which is basically how many spatial modes or principal components that you want to define in your sensor space, basically. So it uses singular value decomposition to estimate these principal components. Generally, most of the physiologically relevant signal is encompassed in the first few modes of the data, and then the latter modes generally contain noise. So what you might want to do is run your DCM with the default number of modes, which is 8 in this case. Have a look at the data in mode space, which I'll show you how to do in a second, and then kind of visually examine which of the modes contain the signal and which of the modes contain most noise. And then you can go back and rerun your DCM with a fewer number of modes. Obviously, the less modes you have, the quicker the model will converge because there's fewer parameters. So it's better to kind of use the default option to make sure that you don't, lose any of your signal, and then see how many modes capture the most data, and then work from there. Like I said, it can often be a trial and error process to really get a model that works best for your data. So those are those options. What you can do with the display window, let's just say I'm going to pick subsample 4. If you look in the display window, I'm going to click display again, and you can see that the data has slightly changed because this is post subsampling. So you can really play around with these different options and see how they affect the data that's being inputted into your model. So to move on to the next section, I'm going to click on this arrow that's here. And there's arrows because each of the different sections in the DCM processing window have dependencies on the previous section. So, for example, you can't start completing the section for the electromagnetic model if you're missing bits from data and design because of these dependencies. So you can move back and forth through the different sections using these arrows. You can also go back if you change your mind or you forget something in the previous section. But that's why you have to use these arrows, basically, because of the dependencies. So, moving on to the electromagnetic model, which kind of allows the specification of these spatial forward models. This is where we're going to put in the locations of our sources that we've identified. So in the left-hand text box, that is where you put the names of your sources. And in the right-hand text box, this is where you put the MNI coordinates of your sources that you've selected. So you can just type them out, for example, like something else like that. Or you can do the easiest thing, which is to load in a map file of the coordinates. So in the GitHub repository, I have a file called example MNI coords. And this is just a matrix of the MNI coordinates. So I'm going to open this. And as you can see, there's 6 rows and there's 3 columns. The 3 columns are obviously the X, Y, Z MNI coordinates in 3D space. And each row is for each one of the sources. So if we go back to our source diagram, we have sources 1, 2, 3, 4, 5, and 6. And each of these 6 rows corresponds to the MNI coordinates of each of those 6 sources in 3D space. So to put our names of our sources in here, we're going to put the left occipital gyrus, the right occipital pole, the left inferior temporal gyrus, the right inferior temporal gyrus, the left inferior frontal gyrus, and the right inferior frontal gyrus. There we go. Okay. Then we have an option here where we can select how to spatially model our evoked responses. We have the imaging solution, which basically applies a patch on the cortical surface. We also have equivalent current dipole, or you could go with the LFP option if you want to model each of your channels as an individual source itself. But we're just going to go with the image option. We also have 2 options here. We have onsets, which basically, if your stimulus is presented at 0 milliseconds per stimulus time, this is the time point where we expect the cortical area to be activated in response to that stimulus. So generally, visual stimuli, you would see the first large deflection after about 100 milliseconds. So we're going to keep this at the default of 64 milliseconds to make sure that we really capture that first large deflection. And then for duration, this refers to the kind of the duration of the input volley, basically. So if you want to vary the width of that and you want to kind of really model the input structure in a much more detailed way, then you can play with that parameter there. But that's not something that we're interested in today, so I'm just going to leave it at the default value of 16. Now, if I click this button dipoles, what happens in the graphics window is we can see the location of source number 1 in the 3D space. This is a really good sanity check for making sure that your MNI coordinates are correct. So source number 1 is our left occipital gyrus, and we can see in the 3D model it's in the left occipital region, which is thankfully where it's supposed to be. So this is a really good way of checking that sort of thing. You can see in this green box here, it's dipole number 1. And then with this drop down, you can select, like this is region number 3, so region number 3 is the left inferior temporal gyrus, and we can see that our yellow dot is exactly where we'd expect it to be in the left temporal lobe. You can see all of your dipoles at once as well. So now we have 6 yellow icons, and we can drag the crosshair around to see if all of our sources are where we expect them to be. Okay, so now we're going to move on to the neuronal model section. And in this section, this allows us to specify the connections in our model that I mentioned when I was drawing out the A, B, and C matrices, and then we can invert our DCM. So here we have excitatory, inhibitory, and mixed. Now, these are referring to the forward A matrix, the backward A matrix, and the lateral A matrix. Now, the reason that in this case it's referred to as excitatory, inhibitory, and mixed is because we've selected the NMDA model, and in the NMDA model, the way that the forward, backward, and lateral connections are parameterized is that in the forward connections, the excitatory pyramidal cells are projecting in the source region to excitatory spiny stellate cells in the target region. So that is an excitatory connection. In the backward connections, there's a connection from the pyramidal cells in the region that the connection originates from to the inhibitory interneurons in the target region. So that is an inhibitory connection, and they are the backward connections. And then the lateral connections, I've already mentioned that we're not going to define lateral connections in this model, but if you were to, then they would originate from the pyramidal cells in the source region, and they project to all of the 3 different cell subpopulations in the target region. So the inhibitory interneurons, the spiny stellates, and the pyramidal cells as well. And that's why it's referred to as mixed. But in terms of mathematical form and role, they are functionally the same. So if we go back to our slide here where we can see our matrices, we have our forward connections from regions 1, 2, 3, and 4 to regions 3, 4, 5, and 6, and then our backward connections from regions 3, 4, 5, and 6 to regions 1, 2, 3, 4. So we're just going to replicate this in these radio buttons here. 1, 2, 3, 4. 1, 2, 3, 4. Okay. And then for our B effect matrix, we have both the forward and the backward connections that we're putting modulatory connections on. So we can estimate parameters about how our task condition might affect these connection strengths, and then also the self connection as well. And like I said earlier, if we look at the A matrix, the buttons on the diagonal are blanked out because self connections are automatically already a part of the model. That's why you don't define them. So I'm going to add this to the B matrix. There we go. Okay, and then finally our C vector. So if you had a more complex task, let's say if you had both a visual and an auditory input in every trial, then this would be a C matrix, and you would have multiple rows depending on the different inputs that you have. Because this is a very simple task, we just have a single vector for our inputs. And we want to define subcortical input, specifically visual input, in regions 1 and region 2. So we just select those. And then finally, we just have some options down here. I'm not going to select any of them today, but I'll just explain what they are. We have the dipolar symmetry option. So this would impose a symmetry prior on your data if you want to model bilateral sources. We also have optimized source locations, excuse me, and this relaxes the zero variance priors on the source locations that you've identified here. You can also lock trial-specific effects, which enables you to enforce all of the trial-specific coupling gains to be the same. And then we also have trial-specific inputs if you want to impose trial-specific C parameter estimation. Like I said, I'm not going to use any of those today, but there they are in case you want to use them. Okay, so usually I would click invert DCM. The DCM inversion process does take some time, so I'm not going to invert it in real time because it would take longer than I have for the tutorial. So I have cooked up some DCMs that I made earlier, and I will show you the results from those. But once you're ready to invert your DCM, you press that button that says invert DCM, and it will open a window with the kind of predicted model outputs and then the actual data outputs. And it will go through some expectation maximization iteration steps that you'll see in the command window. And it also shows the predicted versus actual free energy differences as your model converges, basically. So I'm going to close this and go back to my SPM main menu and click DCM again. Okay, and what I'm going to do this time is I'm going to click load and go to my DCMs, and I'm going to click my full model example. Thinking about it. Yep. You don't want to do this, do you? Okay. I've got to add it to the path. Classic. Classic SPM. Yeah, okay. There we go. Just needed to be in the right directory. Yeah, general rule when you're working with SPM. If something fails first time, check your directories, check your paths. Have you added the path, yes or no? Try again. So we've loaded in our DCM. This is exactly the same as the one that I've just showed you how to define and estimate. But because this DCM has already been inverted, we now have different options to examine the results. So we have this drop-down menu at the very bottom here that currently says ERP's mode. So if we click that, in our graphics window, we can see the model fits in mode space. So at the bottom in this legend here, trial 1 basically means condition 1. So this is the novel image condition. And we have the predicted, so the model estimates with the solid line, and then the actual data in the dotted line, and then same for condition 2, which is the repeated condition in red. So we can see the vast majority of the physiologically relevant signal is kind of captured in the first 4 or 5 modes, really. You might say that modes 7 and 8 probably contain predominantly noise. So what you can do is kind of go back and change the number of modes to say 5 or 4, rerun your model. It will obviously run quicker, and then you can see how the model fits the data in that respect. That's the modes. We can also look at sources. These are the results in source space, looking at our 6 different sources. Once again, the blue lines represent the novel image condition and the red lines represent the repeated image condition. And here we have our 3 different cell subpopulations. So if we go back to this slide here, with our numbers here, cell population 1 was the spiny stellate cells, cell population 2 was the inhibitory interneurons, and cell subpopulation 3 were the pyramidal cells. So that's what's reflected here. It's good to write those numbers down because when you look in the results window, it just doesn't help. It's not friendly. It's like, oh, it's just population 1, and you have to try and guess what that is. So it's good to kind of keep a mental note of those things. We can also look at the coupling, the conditional estimates of the connection strengths in our A matrices that we've identified. So it says coupling A, which means that we're looking at the connections defined in our A matrices. Obviously, the lateral heat map is blank because we didn't define any lateral connections, but we can see the connection strengths and then posterior probabilities on the bottom of those connections that we've defined. We can also look at this in the context of the B matrix. So these are our modulatory connections that we defined in the model, with once again the conditional estimates and the posterior probabilities, and the same for the C matrix. Conditional estimates on the left and posterior probabilities on the right. Trial-specific effects. What we have is the trial-specific conditions for each of the different connections. and the respective connection strengths. So where it says trial 1 and 2, it really means trial condition 1 and 2, so the novel and the image, like the repeated image condition. We can look at that. The input is just basically a gamma function of the input structure. And then we have the response. So this is basically projected back into sensor space. So each of these lines is one of the 64 EEG channels. So we have the observed response in the novel condition on the top and the repeated condition on the bottom, both for the observed data and the model prediction. It's the inverted model, and then that's back projected into the sensor space. And then you can just review, it's the same information just viewed slightly differently. So rather than, if we go back to response, rather than time being on the x-axis and amplitude being on the y-axis, we now have channel number on the x-axis, time on the y-axis, and then the colour in the kind of grey scale heat map is the amplitude of each of those deflections, basically. So in a nutshell, that is kind of how you review the results on an individual level of somebody's DCM, which is kind of useful if you're looking at it in a more exploratory fashion. If you're looking at a large group of people, you might want to run something called a parametric empirical Bayes analysis, which is basically, to put it in very, very simple terms, it's kind of like a Bayesian ANOVA for DCM kind of thing. So you would look at all of your participants that you have. Now let's say you have multiple participant groups, like healthy controls or a patient group, and you can include covariates into your parametric empirical Bayes model to see if any of these connection strengths differ between your group. You can add things like accuracy scores or a reaction time as a covariate to see if there's any correlations between connection strengths and behavioral variables, basically. So people kind of use parametric empirical Bayesian analyses for this sort of thing once you've run your DCM on multiple subjects. What I'm going to do now is, because I have 6 minutes, that is enough time to show you how to run a Bayesian model selection. So like I said before, what you can also do is you can play around with these parameters in the kind of DCM model building environment and see which of the models that you've run on the same data best fits that data, which has the highest model evidence. So I'm going to show you how to do that. I'll close these windows. So one way to do it is to go into the DCM window, and there is a button at the bottom that says BMS, which stands for Bayesian Model Selection. Just to spice things up a bit, I'm going to show you how to access that from the batch editor. If you use SPM a lot, the batch editor is fantastic. I'm going to click the batch button here. I'm back in the SPM window and it's at the bottom. I'm going to click the batch button and this opens the batch editor. Now at the top we have File, Edit, View, SPM and Basic I/O. I'm going to click on SPM and I'm going to go to DCM and I'm going to go to Bayesian Model Selection and I'm going to go to Model Inference. There we go. Okay, so I'm going to specify the directory. So when you run a Bayesian model selection, it will automatically save a BMS file once you've run that comparison. And it's just asking for the directory that you want that file to be saved in. So I'm going to click specify. And I've already made a directory ready for this, so I'm going to click BMS. And then this little dot here just means that I'm going to pick this directory. And now that I've clicked there, it's appeared here to say that that is the directory that I've selected, and I'm going to click Done, which is here. There we go. Now I'm on the data option. I want a new subject. I'm going to click on my subject and I'm going to click New Session. So using this pipeline, you can compare multiple sessions from multiple subjects and compare their different models, basically. So here is where I load in my DCMs. If you're comparing across multiple sessions and multiple subjects, you will need to load in the different models that you're comparing in the same order across all of your subjects and sessions, because SPM is not going to know which one's which. It just sees it as model 1 and model 2. So if you're doing multiple subjects and multiple sessions, just double check that you are loading those models in the same order consistently across everyone. So we're going to specify these models. So I'm going to click Full Model Example, which is the one that we just looked at. And yesterday I was a bit exploratory, and I ran a second DCM where instead of in our modulatory B matrix where we employed modulatory connections on all of the forward and backward and self connections, I only specified modulatory connections on the forward connections. So we have a much reduced B matrix in this second model. So I'm going to compare the log model evidence of these two models, the one that we've just seen and the one with the reduced B matrix, to see which one best fits the data. So I'm going to load that in here. So you can see that I've got the full model first and then the partial model second. And I'm going to click Done. And then for inference method, I'm going to pick fixed effects. Now, if you are comparing models across multiple subjects, I would not recommend clicking fixed effects, because fixed effects uses something called a group Bayes factor, and this does not really account for group heterogeneity or outliers very well. It assumes that all of the models that you're comparing can kind of come from the same model, basically. So if you are comparing models within one subject, which is what I'm doing today, then fixed effects is fine. But random effects is much better if you're comparing models across multiple subjects, because it kind of accounts for this group heterogeneity and assumes that the data in the models from multiple subjects are just coming from a Gaussian distribution. There is a great paper by Klaus Stefan about the kind of fixed effects versus random effects methods. But obviously I have limited time, so that's all I'm going to say. I'm picking fixed effects today because I'm looking at one subject. If I were looking at multiple subjects, I would pick random effects. That's that. And then the remaining options I'm just going to leave at the default. Okay, and then I'm going to click the green arrow here, which is going to run this process. Okay, and we have a plot here, which shows us the log model evidence and the posterior probabilities of our two models. Remember, the first model that I loaded in was the full model example that I worked through today, and the second model is the one with the partial B matrix, which only had modulatory connections on the forward connections. And as we can see here, that second model actually fits the data a lot better than the first one. Obviously, it's only a comparison of two models across one subject, so it's not super informative. If I were looking across the whole data set, I would run this Bayesian model selection across the whole data set. Because like I said, it is only just one subject and it's not super informative. But this is just a quick example on how you run that Bayesian model selection. Okay, so that is all I have to say. I am noticing that it is exactly 4 o'clock. I'm going to say thank you for listening. Here's my contact details and my social medias and stuff. If you have any additional questions, or if you want to hear more about DCM or anything like that, I'm more than happy to answer questions. And yeah, that's it from me. Thank you very much.